To start off our discussion with the oxygen-hemoglobin dissociation curve, we actually have to understand what the function of hemoglobin is. Now, if you ever taken anatomy physiology or you know like what hemoglobin is, a background example or some background information, hemoglobin is going to be a protein that allows for efficient oxygen transport in the blood, either by cooperative binding or releasing that oxygen in tissues. So hemoglobin is necessary for our red blood cell. So we'll just have hemoglobin right here. Let's just say this is our red blood cell. And this is where O2 binding occurs. And we obviously, our red blood cells need to be able to bind oxygen, transport around our body. And we have this protein hemoglobin to help us do that. So that is the function of hemoglobin. And keep that function in mind because this function directly shows us why our oxygen hemoglobin dissociation curve is actually sigmoidal. So I'll do something like this, and that'll show sort of sigmoidal model right there, sigmoidal curve. And obviously with our oxygen hemoglobin dissociation curve, we need to know that our y-axis is going to be our hemoglobin saturation in a percentage form, and over here is going to be our partial pressure of oxygen. All right, so now that we understand that, we have to ask ourselves, well, why is the oxygen hemoglobin dissociation curve sigmoidal? Well, this comes down to the vocabulary term called cooperativity. Specifically, we have positive cooperativity. So in this example, the understanding of a positive cooperativity in hemoglobin is, let's say, let me get my hemoglobin back out here. Let's say this is hemoglobin right here, and let's do this as oxygen. So we obviously know that hemoglobin is going to bind to oxygen. But with positive cooperativity, every one of those oxygens that is binded increases hemoglobin's affinity for oxygen. So kind of just makes it easier to bind more oxygen. So maybe bonding that first oxygen was hard, but after that one, it just gets easier and easier until, we can be in full saturation, so right over here. So that is positive cooperativity and cooperativity in general when we're talking about our hemoglobin. So keep that in mind as well. And another thing that we have to understand is our P50. So let's just say this is like, let's say this is 50% saturation. And our P50, let's say it's somewhere right here. So we're at 50% saturation right here. And our P50, so I'll write P50, this is going to be the partial pressure of oxygen in which hemoglobin is 50% saturated. So this is the partial pressure at which our hemoglobin, 50% of our hemoglobin, is bonded with oxygen. And P50 is very important to know because if you guys look back at enzymes, if you're ahead of that, then for enzymes, we have Michaelis constant, Km. Km is going to be the version of P50, but with enzymes where we're basically going to have the amount of substrate concentration in which we reach half Vmax. But just understand this P50 value, and we also have to know its relationship with affinity because our P50 value really shows us and helps us understand what our affinity is. Now, if we have a high P50 value, this basically means we need a lot of partial pressure in order for our hemoglobin to be 50% saturated, which means that we have a low affinity for oxygen. Affinity. And vice versa, if we have low P50, we have high affinity. The reason why is because we don't need as much partial pressure in order for our hemoglobin to be 50% saturated. So I'm going to keep this relationship right over here when we go through our examples of what can increase and decrease P50, just to have that as sort of reference. And let me delete some of these things over here, and let's actually talk about what can shift our sigmoidal curve from the right or to the left. So I'm going to copy this, move it a little bit ahead, and let's say we see a right shift. So let's say our curve moves to the right. Now, if you remember, and I don't know why I erased this, but let's just say for this entire example, let's say this is our 50% saturation, so P50 will be right over here, so I'll just put P50 for this guy right here. And all right, so we're saying that this is a right shift. I'll put R shift. So when we move to the right, when we're shifting right, we're actually releasing oxygen. So let me move this, and over here we are going to be releasing O2. Releasing O2. And one thing that you'll notice is that our P50 value actually increases. So this was our original P50 value, like maybe 20, and over here we've increased it to 30 milligrams per mercury. So by increasing our P50, we talked about that decreases affinity. So by doing so, when we increase our P50, move to the right, we're decreasing our affinity for oxygen, which means we are going to be releasing oxygen. So it makes sense that when we have a right shift, we are releasing oxygen. Now, what are some, conditions, or what environment could we be in which we have a right shift? Well, one of them, let me change my color over here. One of them is increased CO2. So for example, it's hypercapnia as a medical term, but let's say that our muscles are working, like we're in exercise. So when we're exercising, there's going to be a higher accumulation of carbon dioxide. And if we remember from our bicarbonate buffer system, that it's actually one of the most important buffer systems we have in our entire body. And I'll do this, HCO3 minus plus H plus. We have our bicarbonate buffer system over here. And if we increase the amount of CO2 by Le Chatelier's principle, we need to increase the amount of hydrogens. So with increased amount of CO2, we see an increased amount of hydrogens. And if you guys remember what pH is defined by our concentration of these hydrogens or protons. And if we have an increased amount of hydrogens over here, that means we are going to decrease our pH. So when we are decreasing our pH, we have an increased amount of hydrogen ions, or just protons in general. And another thing that you have to note with our hemoglobin is that we have the T state, and we have the R state. So just some basic characteristics. Our T state, you can think of it as the tense state because we have low O2 affinity. I'll put AF affinity. We have low oxygen affinity in our T state because we're in the tense state, but with our R state, we are relaxed. You think of it as this, relaxed. And we actually have high oxygen affinity. So these are just conformational changes that can occur with our hemoglobin. So over here, this is our T state at the bottom right here. But then when we get to positive cooperativity, which is you see this sharp rise, that's because of our positive cooperativity. When our oxygen binds, this results in higher affinity for oxygen. So we basically increase our possibilities of binding that oxygen until we get to our R state. So remember, T state is the tense state, R state is the relaxed state. Now, another characteristic you have to understand is that hydrogen ions over here, they stabilize our T state. So I'll put over here, H stabilizes the T state. So if we have a high amount of hydrogens, this is going to stabilize our T state, preventing us from getting to that R state. So this is where we see lower oxygen affinity, and this is why we start releasing oxygen. So that's one of the examples that we might see in which, and this is Bohr's effect in it of itself, where we're seeing amount of increase of carbon dioxide, increased amount of hydrogen, decreases pH, and this stabilizes our T state. Now, what about 2,3-BPG? So 2,3-BPG is another molecule that we have to know, and 2,3-BPG, this is going to stand for 2,3-biphosphoglycerate. So let's just say 2,3-BPG, which if you seen a bit, are familiar with glycolysis, then this is something you might be familiar with. And with 2,3-BPG, this binds to the T state. This binds to our T state, or another name for our T state is deoxyhemoglobin. So 2,3-BPG binds to our T state, and it basically stabilizes our T state as well, preventing oxygen binding, and this just lowers oxygen affinity. So over here, I'll do lower O2 affinity. So if we have high amounts of 2,3-BPG, well, this is going to shift our curve to the right, decreasing our oxygen affinity. So these are two conditions in which we might see a decreased amount of oxygen affinity and an increased amount of P50. And when we're in a working muscle, these are the conditions that we might see. So if our muscles are working, if we're exercising, we're lifting weights, well, obviously our hemoglobin is going to dump that oxygen into our tissues because we're using that oxygen to create ATP, and a byproduct of the Krebs cycle is CO2. So this is a constant process that, as you can see, it's integrated throughout our entire bodily systems. But just to keep it simple for today, we're just going to be talking about the oxygen hemoglobin binding curve. So we understand the right shift. And if you guys want to take some notes, you can just pause the video and just take some notes there. But what about our left shift? So now let me just delete all this stuff over here. So I actually want you guys to pause this video and think about it. If we are shifting to the left, what would happen to our P50? Would it increase or decrease? And if so, then what would happen to our affinity as well? So let's say we moved to the left, and let me cut this guy right over here. I'll just leave it at that. But as you guys can see, this is going to be a left shift. So I'll put L shift. And if you said that our P50 is going to decrease, affinity for oxygen is going to increase, then you are correct. So when we have this left shift, we basically lock in oxygen. So we're increasing our affinity for oxygen, so we're binding to oxygen more tightly, and we're able to hold oxygen. So when we're in our left shift, what are some things that can cause this? Well, it's the opposite of everything we just said for our other conditions. So we said an increased amount of CO2, increased hydrogen, which decreases affinity. Well, if we have a decreased amount of CO2, we're going to have a decreased amount of hydrogen ions. This increases our pH, and by doing so, we don't stabilize the T state, and it allows for our R state to occur right over here with our positive cooperativity. And another thing would also be 2,3-BPG. If we have, let me change my color to that green. If we have less 2,3-BPG, well, then our T state won't be, stabilized. We won't prevent our T state from turning to our R state. So our 2,3-BPG, when there's less of that, then we're able to increase our affinity for oxygen and decrease our P50. So that's how we can talk about our left shift. Left shift, when we're moving this way, decreasing our P50, increasing our affinity for oxygen. So what about fetal hemoglobin? So let's just model that as FHB. Well, with our fetal hemoglobin, we are going to have alpha 2 gamma 2 subunits versus our adult hemoglobin. Let me just move it right here. Adult hemoglobin, we are going to have alpha 2 beta 2 subunits. Now, the reason why this is significant is because our beta 1, beta 2 subunits, so I'll just do alpha 1, beta 1, alpha, or this should be beta 2, alpha 2. So this is, what was that over here? I thought I drew 1 right there. So that's 1 right there. This is what our hemoglobin is going to look like, because our hemoglobin is a tetramer. And with our fetal hemoglobin, we're going to have our gamma subunit. Now, the reason why this is significant is because 2,3-BPG binds to beta 1 and beta 2 subunits. And because we don't have any beta 1, beta 2 subunits, 2,3-BPG can't bind to fetal hemoglobin, which results in an increased amount of affinity. It's still sigmoidal, but as you can see, we have a lower P50, higher affinity for oxygen. And a simple way that you can remember this is think of like fetuses or babies in general. They need more of pretty much everything. They need more attention, they need more care, they need more time, and they need more oxygen. So just remember that with fetal hemoglobin, we do not have beta subunits, we have gamma subunits, and this results in an increased amount of oxygen affinity.